Hello guys, welcome to the ninth and the second last lecture in the Life Sciences Cell Biology series. I am Richa, your mentor, and today I am going to talk about a very important cell organelle, that is mitochondria. Now, as you all are aware, mitochondria is a double membraned organelle. It is usually called the powerhouse of the cell. Now, why is it called the powerhouse of the cell? Because it produces ATP for the cell's energy needs. Mitochondria are found in almost all eukaryotic cells, but they are not found in certain cells like the mature RBCs. The mature RBCs lack mitochondria. So, if the mature RBCs lack mitochondria, how does the RBC produce ATP? Where does the ATP come from in RBCs? We know from our knowledge that ATP is produced from glucose. So, three processes are involved in carbohydrate metabolism. One is glycolysis. The next is TCA cycle, and then comes the electron transport chain. So if mitochondria are not present in the RBCs, TCA cycle and electron transport chain cannot occur. So the only process by which ATP can be produced is glycolysis. Glycolysis occurs in the cytoplasm. So ATP in mature RBCs is only produced by the process of glycolysis. This can be a very important question in the exam. So mitochondria has its own circular DNA. So it means that mitochondria can produce its own RNA. It can translate its own proteins. However, not all the proteins required for the mitochondrial function are produced in the mitochondria. Most of the proteins are still produced in the nucleus, and some small amount of proteins are produced in the mitochondria from its own circular DNA. I just told you mitochondria is a site of TCA cycle, that is the tricarboxylic acid cycle or the Krebs cycle, and the electron transport chain. Coming to the structure of mitochondria. Mitochondria is a double membrane organelle. So here in the figure you can see it has an outer membrane and it has an inner membrane. And the space between the two membranes is called the intermembrane space. Now, mitochondria has two aqueous compartments. One is the matrix, similar to the structure of chloroplast. Here also in mitochondria you have an intermembrane space which is fluid filled, and you also have the matrix which is fluid filled. So two aqueous compartments are there, the matrix and the intermembrane space. So the matrix is the interior of the mitochondria, which has a gel-like consistency due to high concentration of water-soluble proteins. So here in the figure you can see this is the matrix. Then you have the intermembrane space, that is the space between the inner and the outer membrane, and that is also fluid filled. Now the inner membrane in case of mitochondria is invaginated to form folds. Here in the figure you can see there are several folds in the inner membrane. So these folds are called cristae. Now what is the role of these cristae? We are just going to talk about it in a moment. In the figure you can also see there is a thread-like purple strand here in the matrix. Now this strand is the mitochondrial DNA. It is visible as a strand-like structure here, but usually the mitochondrial DNA is circular in nature. The mitochondrial DNA occurs in circular form, and these the structure which holds the mitochondrial DNA is called a nucleoid. Coming to the mitochondrial membranes, the outer membrane is composed of 50% phospholipids, and it contains a variety of enzymes for several different processes. So processes that occur in the outer membrane with the help of the enzymes present in the outer membrane are tryptophan degradation, fatty acid elongation, and oxidation of epinephrine. So these are a few examples of the processes occurring in the mitochondria wherein the enzymes of the outer membrane of mitochondria are required. But let me remind you that there are several different processes like this. Now, this process is very important, this reaction. So, please remember that fatty acid elongation occurs in the outer membrane of mitochondria. Now, the outer membrane also has protein-based pores that are big enough to allow the passage of ions and molecules as large as small proteins. So, how do small proteins enter inside the mitochondria? They enter the outer membrane of the mitochondria. They translocate through the outer membrane of mitochondria through these broad, big protein-based pores. In contrast, the inner membrane has much more restricted permeability. That means it does not allow most of the ions or most of the molecules. It has a restricted permeability and allows only certain types of molecules, similar to the plasma membrane of the cell. Now, the inner membrane is divided into two domains. The inner boundary membrane, let me draw it for you. So, this is the outer membrane. This is the inner membrane. And this is the outer membrane. Mind you, these are not two membranes. This is just one membrane. I've drawn it in a way that it is looking like two membranes, but it is one membrane. This is one membrane, the inner membrane. This is one membrane, the outer membrane. Now, the outer side, the side of the inner membrane towards the outer membrane is called the inner boundary membrane. This side is rich in proteins responsible for the import of mitochondrial proteins. So, somewhere in the cell, from the Golgi bodies, the proteins are secreted and they travel to the outer mitochondrial membrane. They then translocate through the outer mitochondrial membrane near the inner membrane. Now, from this part of the inner membrane, these proteins have to enter the matrix. How are they going to enter? There are certain proteins on the inner boundary membrane which lead to the translocation of these proteins directly into the matrix. I hope you are understanding. If you are not understanding, we are going to discuss this in detail in a few more slides. Now, the inner part of the inner membrane, this side, this is called the other domain of the inner membrane. This is invaginated and it forms folds called cristae. Now, why is the inner membrane invaginated? Can you think of something? This invagination increases the surface area to house the machinery needed for aerobic respiration and ATP production. That means that it increases the surface area onto which the enzymes of the TCA cycle and the electron transport chain can be harbored. So due to more surface area, more enzymes, more space to house the machinery or the enzymes or the proteins or the cofactors required for aerobic respiration. That is the TCA and electron transport chain. Basically, the TCA occurs in the matrix and electron transport chain occurs in the inner mitochondrial membrane. The inner membrane is devoid of cholesterol, but it is rich in an unusual phospholipid called cardiolipin. This cardiolipin is a very unusual phospholipid which is only found in the inner membrane of the mitochondria, and this is very important. It has roles in the electron transport chain. It has several roles in the mitochondria and ultimately the cell. So cardiolipin is a four fatty acid chain molecules. You can see here there are 4 chains of fatty acids. 1, 2, 3 and 4. Let me just remove everything. So cardiolipin has 4 fatty acid chains. 1, 2, 3 and 4. It's a tetraoleoyl species. Now the roles of cardiolipin include it stabilizes complex 3 and 4 enzymes in the mitochondrial ETC. It also anchors the enzymes which are responsible for the ETC onto the inner mitochondrial membrane. Cardiolipin is also required for the synthesis of iron sulfur cluster and certain cofactors which are required for these metabolic pathways, that is the TCA and electron transport chain. Further, it is involved in the transfer of protons which is required for ATP synthesis. Then it is also involved in the induction of apoptosis via the release of mitochondrial cytochrome C. The entire process of apoptosis we are going to study in other units, but here you just need to know that cardiolipin has a role in the release of mitochondrial cytochrome C, which ultimately leads to apoptosis of the cell. So these are the 5 roles of cardiolipin. Now the inner membrane has proteins with 3 types of functions. First, I just told you the proteins that are required to carry out the oxidation reactions of the respiratory chain, that is the electron transport chain. Next, it has an ATP synthase complex, which is nothing but the F0, F1 particle, which is required for ATP synthesis. Next, it has specific transport proteins that regulate the passage of materials into and out of the matrix. Now, these materials can be anything from ions to proteins to several other small molecules. Now, the composition of the mitochondrial matrix. The matrix contains enzymes, ribosomes, and DNA molecules. These are circular DNA molecules which are also called the mitochondrial DNA molecules. These are ribosomes specific to the mitochondria, and these enzymes can be enzymes of the TCA cycle, enzymes required for the functioning of the mitochondria, and so on. So the DNA is circular and is present as structures called nucleoids. This I've already told you. The matrix contains DNA that encodes 13 polypeptides in humans. So the mitochondrial DNA encodes only 13 polypeptides. Do you think only 13 polypeptides are needed for the mitochondrial function? No, it's not so. Several polypeptides are needed for the mitochondrial function, out of which 13 are synthesized by the mitochondria itself. Rest all the polypeptides are synthesized or encoded in the nuclear DNA. The nuclear DNA encodes these polypeptides. They are translated on cytoplasmic ribosomes, and then they are secreted into the mitochondria. It also encodes 2 rRNAs and 22 tRNA molecules. So the mitochondrial DNA encodes 13 polypeptides, 2 rRNA molecules, and 22 tRNA molecules within the organelle. These rRNAs and tRNAs are required for mitochondrial protein translation. Now, coming to a very, very important topic, that is the endosymbiotic theory. Now, in simple words, I'll just explain what this theory states, and later I'm going to explain in detail. So this theory basically states that organelles like mitochondria and chloroplast in a eukaryotic cell are nothing but remnants of a proteobacterium which was engulfed by this eukaryotic cell long ago. So imagine you have a prokaryotic cell which has a nucleoid, it has a cell membrane, and it has cytoplasm. Now this prokaryote grows in size and develops infoldings in the cell membrane. So here you can see certain infoldings which start to develop from the cell membrane and they move towards the nucleoid. Now, as these infoldings grew, they pinched off from the cell membrane. Here you can see that these infoldings are no more attached to the cell membrane, but they further go and attach to the nucleoid. These infoldings engulf the nucleoid, and once when the nucleoid is engulfed by these membranes, it becomes nothing but the nucleus, the eukaryotic nucleus. And this is how a prokaryotic cell transforms into a eukaryotic cell due to the presence of a nucleus and an endomembrane system. Now, this eukaryotic cell further engulfs an aerobic proteobacterium. Aerobic means oxygen using. So, an aerobic proteobacterium is engulfed by this cell. The ability of this proteobacterium to utilize oxygen for energy production is utilized by this eukaryote for its survival. In return, this eukaryote houses this proteobacterium and provides it with nutrition and a place to stay. So this becomes something like a symbiotic relationship, wherein the proteobacterium is providing energy to the cell and the eukaryotic cell is providing nutrients and a place to stay to the proteobacterium. Now further, this proteobacterium manages to avoid digestion by the eukaryotic cell, and it continues to live inside this cell. Now, the aerobe's ability to use oxygen to make energy becomes an asset for the host, allowing it to thrive in an increasingly oxygen-rich environment as the other eukaryotes go extinct. So, since this eukaryote was able to survive in the increasingly oxygen-rich environment due to the presence of the proteobacterium's aerobic ability, this proteobacterium was ultimately continued to be housed in the eukaryote, and it transformed into an organelle called as the mitochondria. I hope you could understand till here. Next, if instead of this aerobic proteobacterium, a cyanobacteria was engulfed by this eukaryote, this cyanobacteria had the ability to photosynthesize. So the ability of photosynthesis of this cyanobacterium was utilized again by this eukaryote to survive, and this cyanobacterium, which was engulfed, transformed into chloroplast. Understood? Okay, let's move on to the next topic. So next comes the functions of mitochondria. Mitochondria has some really important functions, out of which the first one is oxidative phosphorylation or the electron transport chain. It's the last and the most important step in carbohydrate metabolism. It also functions to synthesize certain amino acids and heme group. Mitochondria also plays a role in the regulation of calcium ions in the cytosol, and it is involved in cell death or apoptosis. So apoptosis is triggered by the release of certain enzymes by the mitochondria. We are going to discuss the electron transport chain and all these functions in metabolism when we deal with cell metabolism. Here in this lecture, we are just going to deal with mitochondria, its membranes, its basic structure and its functions. Now, proteins sorting to mitochondria. So all the proteins which are required for the mitochondrial functions, where are they produced, how are they targeted to the mitochondria, which proteins are produced in the mitochondria, since mitochondria also has a translation machinery, we are going to study here. So basically mitochondria has 4 subcompartments for protein delivery. So proteins synthesized in the cytosol can be delivered to 4 different compartments in the mitochondria. These are the outer mitochondrial membrane, the inner mitochondrial membrane, the intermembrane space, that is the space between the 2 membranes, and aqueous compartment called the matrix. So as I already told you, only 13 mitochondrial proteins are actually coded by the mitochondrial DNA, and the rest are coded in the nuclear DNA. So once this nuclear DNA is transcribed and later translated on cytosolic ribosomes, these proteins are tagged with an N-terminal signal sequence in the cytoplasm itself. So these N-terminally tagged mitochondrial proteins, how are they targeted to the mitochondria? We're just going to study. Now, once these proteins are synthesized and tagged with the sequence, mitochondrial targeting sequence, there are certain chaperones in the cytosol which bind to these proteins. These proteins are here called pre-proteins because they are in an unfolded state and extended unfolded state until they are targeted to the mitochondria. So we are going to refer to these proteins as pre-proteins because they are still attached to the N-terminal sequence, signaling sequence, and they are unfolded extended proteins. So these pre-proteins which are synthesized on the cytosolic ribosomes are then bound to cytosolic chaperones, and these chaperones deliver these proteins to the outer mitochondrial membrane. Now the outer mitochondrial membrane has certain protein import complexes called as the TOM complexes. Now these signal sequence bound pre-proteins enter the outer mitochondrial membrane through the TOM pore. They translocate through the outer mitochondrial membrane through the TOM complex. So this is the outer mitochondrial membrane, and you have certain protein import complexes. So the proteins coming from the cytosol enter these TOM complexes and they reach the intermembrane space. These are the TOM complexes. Further, once they are into the intermembrane space, now they have to cross the inner membrane also to reach the matrix or to enter the lipid bilayer of the inner membrane. Now these proteins then are translocated through complexes in the inner membrane which are called the TIM complexes. Similar to the TOM complexes, there are TIM complexes in the inner mitochondrial membrane. These pre-proteins are then transported through the TIM protein import complexes in the inner membrane into the matrix. These proteins then enter the matrix. Instead of entering the matrix, they can also laterally exit the TIM complex and enter the inner mitochondrial membrane. Once into the matrix, these proteins having targeting sequences are then cleaved off by mitochondrial chaperones or the mitochondrial HSP70, that is heat shock protein 70. The targeting sequence, that is the N-terminal sequence which was the mitochondrial targeting sequence, is cleaved off by the HSP70 chaperone present in the matrix. Later, once the targeting sequence is cleaved, there are several other chaperones in the matrix which assist in the proper folding of the protein. Now these matrix targeting sequences, that is the N-terminal sequence that leads the protein into the matrix, are contain positively charged amino acids and hydroxylated ions. So any pre-protein which is to be targeted to the inner mitochondrial membrane will not have positively charged amino acids and hydroxylated ions. Only those pre-proteins which have to enter the matrix will have a targeting sequence with positively charged amino acids and hydroxylated ions. Now the inner mitochondrial membrane has TIM complexes, I already told you. There are two types of TIM complexes in the inner mitochondrial membrane. These are TIM22 and TIM23. Now what are the roles of these two? TIM22 binds integral membrane proteins of the inner mitochondrial membrane. So those pre-proteins which have to be integrated into the inner mitochondrial membrane are translocated through the TIM22 pore and they insert themselves into the lipid bilayer of the inner membrane. While TIM23, that is the protein import pore of the inner mitochondrial matrix, that is TIM23, recognizes the matrix proteins. So the proteins which have to enter the matrix are translocated through the inner mitochondrial membrane via TIM23 pore, and they enter the matrix. I know this is very confusing. I am going to explain it once again with the help of a diagram. So here in the first case, this is a matrix precursor protein with a pre-sequence. This is the pre-sequence or the N-terminal matrix targeting sequence, which is going to target this pre-protein into the matrix. Can you see these plus signs here? These denote charged amino acids, positively charged amino acids. So this pre-protein which has to enter the matrix, first it reaches the mitochondria and crosses the Tom complex. Now once it crosses the Tom complex, it is in the intermembrane space. Then it crosses the TIM23 complex. Why TIM23? Because it is a matrix protein, a protein which has to enter into the matrix. Any matrix protein will translocate from the TIM23 complex. Now once into the matrix, mitochondrial chaperones such as MTHSP70, that is mitochondrial heat shock protein 70, comes and cleaves off this targeting sequence as well as assist in the proper folding of the protein. So here can you see, with the help of the chaperone, this protein has been folded into its native matrix protein configuration. Now come to the second case. This is a pre-protein which has to enter the inner membrane of the mitochondria, and it has to become an integral membrane protein of the inner mitochondrial membrane. Similar to the matrix protein, this also enters through the Tom complex and comes to the intermembrane space. Now, instead of going into the TIM23 complex, this will go into the TIM22 complex. Why? Because TIM22 translocates proteins of the inner mitochondrial membrane. Now, it does not cross the TIM22 completely into the matrix, but it laterally exits the TIM22 and enters the bilayer of the inner mitochondrial membrane, and it remains as an integral membrane protein. I hope this was clear to everyone. This was all about mitochondria, its structure, its membranes and its functions. The detailed functions of mitochondria, that is the functioning of the electron transport chain, elongation of fatty acids, all this we are going to study when we study cell metabolism. Now in the next lecture, we are going to discuss certain questions that have already come in the CSIR NET Life Sciences exam, and this lecture will be called the first exam cram. All my lectures dealing with previous year papers, previous year questions, their answers are going to be called as exam cram. So you have to cram these questions. So the next lecture is going to be all about questions on cell biology and the topics that we've just covered in this playlist. Do not miss the next lecture. Thank you. Please subscribe to Utkalp Digital Classes for all the latest content on CSIR NET Life Sciences. Please do not forget to press the bell icon for the latest notifications. Thank you. See you again soon.